Hi, everyone. I'm Faraz Ali. I'm the CEO of Tenaya Therapeutics. I'm really excited to have the opportunity today to tell you more about our company. We are 100% focused on heart disease, and we like to say that we're inspired to change the face of heart disease. Our vision is to transform the lives of individuals and families fighting heart disease and to discover, develop, and deliver curative therapies that address the underlying cause of heart disease. These pictures you're seeing are pictures of actual people who've been affected by genetic heart disease, either genetic hypertrophic cardiomyopathy or genetic dilated cardiomyopathy. And what you'll notice is this is a disease that runs in families. There's entire families affected by this disease in some cases. It's autosomal dominant. And you see that it affects not only adults and, this is not heart disease of your grandparents. This is heart disease that also affects children, as you can see with the pictures on the far right. So we're inspired to try to advance curative therapies to help people like this. Our strategy, in addition to being 100% focused on heart disease, is to pursue prevalent and orphan indications. And so we have a bit of both in our portfolio. We're modality agnostic, so a lot of what we do is gene therapy, but we actually also have a small molecule approach that we're advancing. And we like a precision medicine approach where we like to test drive the science, efficacy, and safety in an orphan population before moving into the broader prevalent population. Very strong foundations, great founders. We raised more than $140 million to date in the Series A and Series B. We're currently about 70 people, full-time employees in South San Francisco, mostly in R&D manufacturing, and approximately 30% have a PhD, so very strong focus on science. Very strong capabilities, which I don't have time to go through today, but we'll cover some of them. We've made a tremendous amount of progress. We have discovered novel vectors for the heart. We have compelling proof of concept in multiple projects. Again, we won't have the opportunity to talk about all of them today, as well as in-house capabilities for AAV manufacturing. We have turned the biotech playbook on its head. So instead of having one platform that we have deployed in multiple therapeutic areas, we actually have 3 distinct platforms that are 100% focused on heart disease. And we like to say that each of these platforms are solving a different problem. On the top right, regeneration, when the problem is a loss of myocytes, a loss of cells due to a heart attack, or for that matter, COVID-19 infection, there's no therapy on earth that can regenerate those cells, and the only solution is to create new cells. And so we have 2 different approaches for in vivo regeneration. It's not cell therapy, it's gene therapy, but in vivo regeneration of new heart tissue. And we'll talk about that today. On the left, where the problem is in a loss of cells, but cells that are defective because of a mutation, well, the solution is more of the kind of lock and key gene therapy that has become quite familiar as the gene therapy space has evolved over the last 10 years. And so over there, we're focused on genetically defined cardiomyopathies. And on the bottom right, where the problem is finding new targets, and where we've depended on mice and rats and dogs and sheep to find targets, well, the solution, and those haven't been predictive of the human condition, well, the solution is to use human iPSC-derived cardiomyocytes, again, not as a therapeutic, but as a way to model the disease and to find targets and to discover new therapeutics in a modality agnostic way. So we have quite a lot going on, IP, know-how, and capabilities in many different directions. So how are we pursuing this unique strategy? Part of it is by internalizing a lot of capabilities under one roof. So we've uniquely integrated the capabilities both for vectorology, for not only for the heart, but specific cells in the heart, the cardiomyocyte versus the cardiac fibroblast. And the top, you can even see that the vectors we discover depend on not only what cell that you're pursuing, but what specific delivery vehicle you're pursuing. And we have filed a lot of IP on novel capsids, novel promoters, and other regulatory elements. On the bottom left is we use heart disease models for target ID and drug discovery. As I mentioned earlier, we have both 2D and 3D models for this. We have isogenic lines, we have iPSC derived lines, we have patient lines to do that. And on the bottom right, you see manufacturing. So we've internalized non-GMP capabilities, but also we're beginning to pursue GMP capabilities. And we're operating at the 200-liter scale today and moving up before we ever get into the clinic. So all of these have been uniquely targeted for the heart. So I'm going to talk a little bit right now about gene therapy. We have many projects going on right now, and we don't have the time and I don't have the ability to share all of that great work, but we'll talk about one project today within gene therapy. And that's the DWORF gene therapy project. This is a program that we licensed from one of our founders, Eric Olson at UT Southwestern. This is a novel therapeutic. We have unique IP in this space to pursue it. What's really exciting about DWORF, which works on the SERCA pathway, but with a completely different mechanism than prior gene therapy effort on SERCA, is that we have proof of concept for dwarf overexpression in multiple models. And that actually creates multiple horizons of opportunity, starting with an orphan population and all the way to chronic heart failure that affects 1 million people. And so as you can see here from this slide, the initial proof of concept we have is in the phospholamban mutant population that has arrhythmogenic cardiomyopathy or dilated cardiomyopathy. And we have proof of concept in 2 different models there. Then in non-genetic dilated cardiomyopathy, we have proof of concept for AAV DWORF that ameliorates the heart function in that model. And I'll show you a slide or 2 there. But really excitingly, even as you go beyond that into MI and post-MI and HFrEF, we also have reason to believe that overexpression of DWORF can be relevant over there with the gene therapy approach, in part because we have evidence from failing hearts that DWORF is underexpressed. And as DWORF is underexpressed, then SERCA is underexpressed, which decreases the contractility of people's hearts. So some illustrative data, we have an example here of AAV9 DWORF that is increasing the expression when delivered in an IV fashion to a relevant model, in this case, a dilated cardiomyopathy model, the MLP knockout model. We see the high increase of DWORF expression. We see an improvement in heart function, and we see a reduction in adverse cardiac modeling. So these are significant improvements, and actually through dose ranging studies, we're able to get even better improved efficacy. This is data that has been submitted for publication. We've also been able to show that AAV9 plus DWORF efficacy can be established in an acute MI mouse model. And over here, you can see that we're improving the heart function, we're reducing cardiac dilation. Now, we're not improving the scar size, and that actually is expected per the mechanism of action of DWORF. It's going to work on the cells that are alive and not on the cells that are dead. So we're not expecting to see a reduction in scar size. So that's one example of a gene therapy approach that we're pursuing. We have others that, unfortunately, we don't have time to talk about today. Now, importantly for our gene therapy, I mentioned earlier that we've internalized vectorology, and this is an example of an effort. We've internalized the ability to create multiple libraries. We've actually created 100 million variants and have already done initial screens to find, and we have found vectors with higher transduction, higher expression, lower antigenicity, better target cell selectivity, manufacturability, and importantly, intellectual property for both capsids targeted at cardiomyocytes as well as capsids targeted at cardiac fibroblasts. We actually presented a poster on this at the American Society of Gene and Cell Therapy earlier this year, and I don't have time to go through the details of it, but indeed we showed several vectors, in this case with a focus on the cardiomyocyte, several vectors that outperformed AAV9, both in human iPSC-derived cardiomyocytes as well as in vivo models in murine hearts. We see lower antigenicity, better transduction of heart, better heart to liver transduction ratio, so more liver to targeting. And so overall, we're pleased with this early progress. But we're not stopping there. So that was actually our first generation approach, where we started that last year and continuing to this year, 100 million variants from 4 libraries, where we started in human cells, moved to the mouse, and then moved to non-human primates. And we have our early data there that we presented, and more to come in this year. But we've already initiated a second generation effort where we have 200 million variants from 16 libraries. We're doing direct screening on non-human primates. And so we're looking forward to getting data from that in the coming 6 to 9 months. And after that, we're going to pursue yet more approaches after we've learned everything that we can from the first 2 in 2021. So this is a real area of strength for us. And actually, we have received a lot of inbound interest from people on our vectorology efforts, other people who are interested in gene therapy for the heart. Importantly, to go after a gene therapy for the heart, we thought it was important to internalize as many capabilities as we can regarding manufacturing. We're not only pursuing orphan populations, as I mentioned at the top, but we're also pursuing prevalent indications. And to do that, we need to be able to scale up manufacturing to beyond a 1,000 liter, beyond 2,000 liter, but to 5, 10, and even 20,000 liter to bring consistency and to bring cost of goods down to a commercially relevant point. And so for that, we've, at this current moment in time, we've internalized both HEK and SF9, partly in order to do head-to-head comparability of those 2 manufacturing platforms for existing capsids as well as new capsids. But then we're also scaling up to 200 liter. That's enough for IND-enabling tox studies. Importantly, we can produce enough material for large animal studies, not just small animal studies. So for cynos or pig studies. Pig studies are the gold standard for heart disease drug development. So we're already there today. What's emergent and we're excited to share is that we're now beginning to prepare for the possibility of becoming fully integrated from a GMP perspective. So we have filed some IP in the manufacturing space. We've licensed other foundational IP, and we are actually actively seeking to internalize cGMP capabilities. We have 18 people today, and we're growing that every day, that are 100% focused on upstream, downstream, or analytical development and quality control of AAV manufacturing. So this has become a real area of strength for us. Finally, the last part I want to discuss is just the opportunity we have to regenerate new heart tissue with a regeneration platform. And in this instance, what we're doing is different than what we're doing with gene therapy. We're trying to create new muscle. How do we do that? We do it via a direct injection using an AAV that is targeted to the cardiac fibroblast, and in vivo deliver these vectors with proprietary transcription factors or combinations of transcription factors to resident cardiac fibroblasts, and that induces them to reprogram and become new cardiomyocytes in the human heart. We have attempted to demonstrate proof of concept this in the dish with human iPSC cardiomyocytes and fibroblasts. We've demonstrated in multiple murine and rat models, and we've also now excitedly we're excited to show that we have also demonstrated that in large animal models, including the pig acute and pig chronic MI model. Importantly, we have put all of the different pieces together in a single AAV. It's a novel capsid that is derived from AAV5. And there are multiple transcription factors here packaged into a single AAV. We have regulatory elements that limit the expression of these in cardiomyocytes so that the focus is on the cardiac fibroblast. So really a precision approach to deliver the right material to the right cell. And with that, what we have been able to show is some very good results. This is also a snapshot of a poster from the American Society of Cell and Gene Therapy back in May of 2020, where we showed the results in a dish, results in a murine model, and then ultimately the results in a pig. We're very pleased to show a significant greater than 10% improvement in ejection fraction in a chronic post-MI model. And these results actually really, when you compare to other results of other cell and gene therapy approaches, really stand out in a very positive way. This is improvement not against a declining control, but actually true improvement in ejection fraction above a lower baseline following a heart attack. So this is quite exciting results, and we're in lead optimization stage for this project. Finally, very briefly, I can't finish this presentation without also mentioning our precision medicine platform, the use of iPSC-derived cardiomyocytes for drug discovery. We fully integrated all the pieces we need to do this, to do phenotypic screening in human iPSC cells with a focus on heart disease. We've created multiple disease models, both 2D as well as 3D heart tissue. We have multiple assays. We've actually internalized image analysis and machine learning and AI algorithms to be able to process the data. We have multiple screening methodologies and multiple in-house models, and we're able to screen, and we've demonstrated that we're able to screen in a modality agnostic fashion to find targets. This is an overview of what that looks like. We, for example, for one disease model, the isogenic lines, we take wild type cells, mutate them using CRISPR to insert human disease-causing mutations, and then we differentiate them and mature them. And so now we have 20+ lines that represent a large number of the DCM mutations, dilated cardiomyopathy mutations in humans. We also have cells actually from humans that have all the complexity of human genetics, and these are severe patients who experience heart transplants before the age of 30. And so we're able to do discovery in the isogenic lines, but then confirm the results in actual patient-derived lines. It's been very exciting. We've actually did our initial screen about 2 years ago, and through that we actually found multiple hits, and we were very pleased to find that we could use the combination of the phenotype of the iPSC, the mutated iPSC cells, with machine learning algorithms to find hits, and we were able to confirm that these were biologically relevant hits. One example of that is that one of our hits was, for example, the omecamtiv mecarbil compound from Cytokinetics. And so that told us that we were getting biologically relevant compounds or biologically relevant hits through this process. We were able to confirm the targets as well. And so now we're on to next generation versions of this, where extremely high resolution imaging that allows us to characterize these cells in different isogenic lines and in different models to look at sarcomere length, sarcomere density, to look at the length of the sarcomeres, the contractility, and a whole bunch of data. And as you can see from the graph on the right, we can actually use this to discriminate the effect of a target in different mutations, and then also then use that to discriminate the effect of different compounds in these different mutations. So we're able to do this now in a high throughput and high content way to find gene therapy targets as well as small molecule targets. And in fact, the first project coming out of this is actually a small molecule project that we are advancing, and we're in lead optimization stage for that project as well. So with that, I'm going to close. I hope in the short time that we had, we covered a lot of territory, a lot of content, but hopefully you have a sense of the uniquely integrated capabilities that we have put around to the company to be able to pursue this vision and this mission, which is to discover, develop, and deliver curative therapies that target the underlying causes of heart disease. And so we like to start and finish with a focus on patients. We're inspired by these people and their stories, and there are no good options today for most of them. There's generic neurohormonal antagonists or heart transplants, and we think we can do better. And heart disease hasn't been a popular area, but we like to say that the science has arrived, but the crowd has not. And with Tenaya, we're hoping to build a sentinel company that will be the leader in gene therapy for heart disease. So thanks for your attention. And if anybody's interested in learning more, please feel free to reach out to us during or after the conference. Thank you.